which one is typical, which cells are typical, and which cells are atypical. Yes. The specimen is the ovary. The ovary is an organ. The ovary is not the same as an ovum. We are not asking you to draw the entire ovary, in as much as you are going to draw this again, the entire ovary, during the second cell, when we go to female reproductive system. We are asking you to focus on the ovum, the cell. All of these are cells. Cells, follicular cells, making up the follicle. These are cells. All of those are cells, but they're too small for you to study. Even these are cells of the connective tissue. You have primordial cells there, which are too small for you to study. That is the reason why we are asking you to concentrate on this cell. This large cell is the ovum. So the ovum is not the ovary. We are asking you to look at that. So what can you see? What organelles can you see? Aside from the cell membrane, you have a zona pellucida that is similar to a capsule in other cells. It's like a capsule, the zona pellucida. You have the cytoplasm, which appears granular. You don't actually see endoplasmic reticulum or ribosomes, but the most prominent would be the nucleus and one or two nucleoli. Okay, but the side question can ask you what does the nucleus consist of. Okay, so you can see there the cell membrane. No other organelles. You don't see mitochondria, don't see Golgi apparatus, etc. The most prominent here would be the nucleus, in some other specimens, the nucleus and the nucleoli. Next slide. So this is a typical cell. That is a typical cell. The ovum. Next, you have the slide on the spinal cord. Again, we are not asking you to draw the entire spinal cord. Your topic is not nervous system. Your topic is the structure of the cell. You're just wasting your time in the laboratory room drawing everything, when in fact you're going to draw again the entire spinal cord during the second cell under nervous system. So all you have to do is focus on one or two of these neurons, motor neurons. Choose one that is representative of a neuron. You will notice that the neuron is stellate in shape, has numerous cytoplasmic processes. The small cytoplasmic processes are dendrites, and the long cytoplasmic process is an axon. So like that, the appearance of your drawing should be something like this. These are also cells, nucleus of neuroglial cells. These are also cells, neuroglial cells, the nucleus of neuroglial cells. But again, they're too small for you to study. That's why we are asking you to concentrate on the neuron as a cell. What do you see? You see cytoplasmic processes, dendrites, axon. You have a fish-eyed nucleus. If you move the high-power objective up and down, you will see granules in the cytoplasm. These granules are Nissel's bodies. Nissel's bodies correspond to ribosomes. In ordinary cells, the Nissel bodies correspond to ribosomes, not endoplasmic reticulum. Because you know already that the endoplasmic reticulum is a network of tubules. These are bodies. The Nissel's bodies, they are attached to endoplasmic ribosomes. They correspond to ribosomes that are attached to the endoplasmic reticulum. Okay? So this is your neuron. You'll get a more detailed picture of what neurons are like when we go to the nervous system. So we're just asking you to draw, illustrate the large cells where you can make observations, the structure of the cell. Next session is epithelium. You were already lectured during the lecture R on epithelium. Epithelium lines body cavities, lumens, surfaces. Keep that in mind. And having that in mind, that epithelium lines spaces, lines cavities, lumens, the epithelium can be classified into simple if there's only one layer, and stratified if there are several layers. Okay, so where is the epithelium now? Lines the cavity. This is the lumen. The specimen is a blood vessel. This is a blood vessel. We are not asking you to draw the entire blood vessel. You'll go to that under circulatory system. Okay, all we are asking you to draw is the epithelium. This is now simple stratified squamous epithelium, that one, in side view or on edge. What did we see? Some of you drawing surface view. Can you see single layer on surface view? Pag stratified squamous, several layers, ang surface view pareho sa simple squamous. Kaya nga nandyan sa inyong manual, on edge, side view, get a coin. Ay, wala akong coin. Get a coin. Ang on edge, ganyan. So you can see single layer. Anong mga drawing ng iba? Ganyan, and face. Maraming ganyan. Bakit ganyan ang drawing ng iba? Kasi dun sa atlas, ganon. Diba? Pag tignan mo yung stratified squamous, ganon din ang itsura and face. But we're asking you to draw simple squamous, so it's on edge, ganyan. Kaya it's supposed to just appear like a line. So this is the lumen of the blood vessel. These are blood clots. In case you didn't know that this is a blood clot, only that is the simple squamous epithelium. In blood vessels, it is specifically called endothelium. Simple squamous epithelium in blood vessels, it is called endothelium. In serous spaces, serous cavities, it is called mesothelium. So for example, in the peritoneum, in the pleura, it is a mesothelium. So under high power objective, ito lang ang simple squamous epithelium, specifically called endothelium. What were your drawings like? You were concentrating on this. This is the connective tissue. This is the tunica media. If you were concentrating on this, you will see several layers, and it will not be simple squamous anymore. It will be stratified kasi ang daming layer. But that is not the epithelium. The epithelium is that. Okay? Next slide. So this is simple squamous. The specimen is alveoli of the lungs. Alveoli of the lungs. So these are the spaces in the alveoli of the lungs. These are your septa, septal wall of the alveoli. For gas to be able to diffuse, it has to be thin. And because it is thin, it has only one layer. Look there, only one layer. So this is simple squamous. simple squamous. Next slide. Next slide.